Hi guys, welcome to our lessons today. We'll be exploring monohybrid crosses using Punnett squares. Let's start off with some background knowledge and some fun facts from the one and only. Let me introduce Professor Harry Bryant. Take it away, sir.
Hi folks, I'm going to start off talking about the one and only Gregor Mendel. Mendel's observation became the foundation of modern genetics and the study of heredity. His experiments showed that the inheritance of a certain traits in peas plants follow the particular patterns sequence becoming. Mendel chose to use pea plants for his experiments due to their many distinct varieties, and becoming offspring could quickly and easily produce. He cross-fertilized pea plants that had clearly opposite characteristics. For example, tall with short, smooth with wrinkly, those containing green seeds with those containing yellow seeds. Mendel's terminology consists of factors and traits, but after his discovery, the vocabulary on the topic widened to alleles and genes, which we know today. But enough from me, let's head to the classroom for more knowledge.
Well, you had it here first. Gregor the Great Mendel, father of genetics, discovered through his works on his peas the fundamental law of inheritance. He reviewed that genes come in pairs and are both individually inherited from each parent. Mendel tracked his theory to the parents' genes and their appearance in the offspring and conducted that they be both dominant and recessive traits. As pea plants were able to produce rapidly, Mendel was able to perform many experiments focused on the one trait and examine them. This improved his results as multiple experiments of the one was able to be completed many times, making his data consistent and reliable. Don't understand? Well, don't worry, I've got someone here who can help, an expert on Mendel's experiment. Let's get straight into it.
You've heard all the background knowledge, but I've got something that may help you understand. Some of you may have heard the term Punnett squares. Well, I'm going to explain Mendel's work on pea plants using Punnett squares. Let's start off with what a monohybrid cross actually is. A monohybrid cross is a breeding experiment between the pea generation, meaning the parent generation, that differ in a single trait. The pea generation organisms are homozygous for the given trait. However, each parent possesses different alleles for that particular trait. Mendel got true breeds and bred them for 2 years prior to his experiment. A Punnett square may be used to predict the possible genetic outcomes of a monohybrid cross basis on probability. Let's keep in mind this is just probability. Now let's use Punnett squares to broaden our understanding. We'll do a Punnett square on Mendel's study of pea plants. We'll be focusing on Mendel's study of the purple flower and the white flower, both male and female. First, we'll start with a Punnett square as shown, and insert the allele of the male and female flowers as so. Just a reminder, Mendel's vocabulary did not include allele, he used the term factors. How interesting. Okay, back to business. Once you've inserted both alleles into the Punnett squares, you combine the alleles as so, moving the capital B down and the little b across, keeping the big B at the front. Now that we have completed the Punnett square, you can see that the phenotype of the alleles are purple, and the purple gene is dominant, and the genotype is BH. We call this the F1 generation. This is where Mendel discovered the law of segregation, as only one allele from both parents gets passed on to the offspring. But what would happen if we crossed two of the F1 generations? Let's find out. Let's start by filling in the two alleles from the F1 generation, and repeat the process from the F1 generation Punnett square. As you can see, the difference in the allele of the female gene, little b, is expressed more. We can now see the phenotypes and genotypes. 1. Purple phenotype, genotype big B. 2. Purple phenotype, genotype big B, little b. 1. White phenotype, genotype little b. This gives us a 3:1 ratio of purple to white phenotype. Thank you for watching. I hope this video was helpful in explaining monohybrid crosses using Punnett squares, and maybe you can explore your own and discover something new. Maybe become the next Mendel. Don't forget to like and subscribe. Thanks!